All right, should we get on to the next part of the presentation from Paris on larval fish?
Sure. All right. So again, now we're going to sort of switch into, um, talking about fish, but probably not the fish that you guys are are thinking about. Not these certainly aren't trophies. Um, but okay, so larval fish surveys. So that was one of the things that was an emphasis in the 2020/21 CSMI was to do this really spatially intensive larval fish survey. And the reasoning behind that is because Lake Michigan has undergone these significant changes. This is coming from changes in nutrient inputs and invasive species, kind of like what we were just talking about, what the Dreissenid mussels are doing to nutrient dynamics. So we're seeing changes in primary productivity. We're seeing less algae is produced. We're seeing declines in zooplankton abundance and shifts in their composition. So specific types of zooplankton tend to be the ones that are winning out. And unfortunately, we've seen relatively low fish recruitment, natural fish recruitment in the lake over the last decade or so. What we do know also is that the changes are not uniform throughout the lake in space and time. A lot of the information that we get is sort of offshore only um, during like regular monitoring. So we wanted to really like emphasize spatial coverage of the lake for this CSMI survey. And so talking about changes in primary productivity, I actually had a student who looked into this. And so I'm going to step you through all this. But essentially what this student was doing was uh, looking at satellite photos of Lake Michigan and Lake Huron. We're going to focus on Lake Michigan here. Um, but every day there, since the 70s, we're only going to focus on from about 94 through 2017. But every day there's a satellite that goes over and takes a picture of the Great Lakes, takes a picture of Lake Michigan. And working with some really clever people, some optical physicists, they actually figured out that if you look at the ratio of the colors in each pixel from that satellite image, you can actually relate that to chlorophyll on the surface. So it's essentially a measure of the amount of chlorophyll at the surface. Um, and then, so that's what we did. We took all those images and we ran them through these algorithms and we got these maps of surface chlorophyll. And so here I'm showing these maps just to show you, we split everything out by depths. So the colors represent different depths, going from, like the reds into the blues, you're going from shallow water into deeper water. And then if you look over here at these graphs, this is sort of the take-home message here. The graphs are showing on this y-axis we have chlorophyll concentration, and on the x-axis we have years. So it's a time series of surface chlorophyll concentrations that are estimated from these satellite images. And so it's split out into different months. So let's look at this top one here. Figure A, this is May in Lake Michigan. This is the average surface chlorophyll in Lake Michigan during the month of May over this time series. And then each line and each color sort of represents how that's changing with depth. And so if you look at all three of the Lake Michigan graphs, what you see is that we're not actually seeing much change in surface chlorophyll in shallow areas. So that top line, that darkest red line, it's either flat or actually increasing through time. So we might actually be seeing increases in chlorophyll in primary productivity in nearshore areas. But look at this May graph. As you go out deeper, you're seeing this decline through time, and it's pretty striking, and it's pretty consistent. And so offshore, we're seeing decreases in primary productivity. We're seeing decreases in the base of the food web through time. And so this has an effect on food webs all over. And it's also spatially variable. I don't have time really to go through all of that, but just keep it in mind that you're seeing decreases in primary productivity, particularly offshore. We also see, like, if you look at this picture of, or this map of Lake Michigan, if we divide this out into quadrants, we can take some of this data. We can actually use another model to look at how that affects fish recruitment. So this is, this is modeled data. So this is not real data, but it's basically taking a model that's taking all of the different abiotic factors and all of the lower trophic level factors, and it's projecting recruitment of different fish in different parts of the lake. So the fish that actually survive, where are they coming from? That's what we're getting at here. And so here we actually include Green Bay in this one. So Green Bay is this first one, and then we have the two. We have the northwest and the northeast parts of the lake, and then southwest and southeast. And so according to our models, according to the things that we know about the lake, if we put that into a model, what it looks like is most of the fish that are actually recruiting out, most of the fish that make it to the adult stage are coming. There's a lot that are coming from from Green Bay. But then if you look at the main part of the Lake Michigan, most of the recruitment, this is for yellow perch, most of the recruitment is coming essentially from the southern basin of the lake. And that makes a lot of sense. That's like the warmest part of the lake. You also get a lot of nutrients coming in from big rivers like the St. Joe's River. And that's what's affecting, that seems to be where fish are coming from in terms of yellow perch, at least. We did the same model for alewife, and we get really similar results. Again, most of the recruits are coming. There are a lot of recruits that are coming from Green Bay. But if you look at the main part of Lake Michigan, a lot of the recruitment for alewife, according to this model, should be coming from the southern basin. So we have this kind of like regional contributions of different recruits. All right. So, and again, as I mentioned, the whole process of CSMI is that, the managers are actually hearing, from their constituents about what's important, because alewife are important. This is something that was actually a surprise to me, that, when I was sort of learning about the Great Lakes, it was the alewife are this invasive species and it's a problem. But they're become so ingrained in the food web. Dan showed the diet results from Brian Roth. Everything's eating alewife. And so alewifes now, it's an important thing. We need to think about it, really for management. So CSMI was designed around trying to figure out what's influencing alewife recruitment. So alewife are the most common prey for salmon in Lake Michigan, but their biomass has been low over the past decade, and kind of what are the causes. The general hypothesis that we're going with is that it is something that's happening at this larval phase. So shortly after they're hatched, when they're just tiny little threads in the water column, we know their mortality is through the roof at that stage. Like they're 99.9999% of the fish are going to die at that stage. And these tiny little changes in survival at that stage can make a huge difference when you go out and actually think about lake-wide recruitment. So we're doing this larval fish survey in the 2021 CSMI assessment to try to look at what's affecting alewife survival at the larval phase, at the larval stage. And one thing that might be important, and we're looking again at sort of all of the things about the lower trophic levels, because it could be veliger consumption. So the quagga, we know there's a lot of quagga mussels out there, and they release veligers as their like reproductive products. A little tiny, think of these as little tiny baby quagga mussels, and they can be like overwhelming how many of them are out there at a certain time. And so there's actually some people are working on the idea that it might be these veligers, which are new in the system, and we don't know how important they are nutritionally. We know fish are eating them. We know larval fish are eating them. And so maybe that's part of it. Maybe it's part of like the shift in zooplankton composition and what the fish are eating. That's the problem. So this is a pretty interesting picture here. These are 2 yellow perch larvae, one that was fed 100% veligers and one that was fed zooplankton diet, and these are the same age. I believe they're about 20 days old. And so you can see the one that's actually eating zooplankton rather than veligers is growing, it's twice the size. So maybe that's the issue. And so that's why this larval fish survey was designed, to go out and see how alewife larvae are being produced around the lake, and then what are they eating. All right. And so this is what the survey that took place. This is a huge effort. So this was, I believe, 16 transects that were sampled. This is all done during the month of July during 2021. There were 3 large federal research vessels that participated in this survey. The Lake Explorer 2, the top ship here, that's run by EPA Office of Research and Development. And they did the northern transects, so the ones that are in blue here. And then the RV Sturgeon, the blue vessel down here at the bottom, the USGS Lake Michigan boat ship, they did the southern transects. And the Lake Guardian, the EPA ship that I was talking about, they did support on kind of all of these transects, I think. So anyway, so this is a huge effort. 3 ships working in coordination over the span of a month to sample all of these sites. They also sampled different parts of the water column. So one of the other things that's happening with all these quagga mussels in the lake is that the lakes are becoming more clear. So there's less algae in the water. There's more water clarity, and I'm sure some of you that have been on the lakes for a long time have noticed this, that the lakes are becoming more clear. So one of the ideas is that maybe it's actually UV is a source of mortality for larval fish. So alewife are especially sensitive to UV light, and it can be a source of mortality for them. So they actually measured, they collected larval fish and zooplankton from different parts of the water column. So there was this UV zone, which is basically the surface, and then the part of the water column where UV light goes down to 1%, and then the no UV zone. So this is an area where fish are safe from the harmful effects of UV light. And so they sampled up to 3 tows per site, 1 at the surface, 1 through that 1% UV zone, and then 1 down in the no light zone, the no UV zone. So now we'll get into some of the results. This is the results from zooplankton. As I mentioned, they sampled zooplankton at the same part in the same parts of the water column and at the same sites. This graph is showing nauplii from copepods. So these are just baby zooplankton, so the smallest fraction of zooplankton. They also happen to be the most abundant zooplankton that's out there. But just overall, in terms of summarizing what we found with zooplankton was zooplankton communities are dominated by calanoid copepods. So that's a specific type of zooplankton that does really well in cold systems and in systems that are not very productive. So that's not terribly surprising. But again, it just kind of reinforces that these are cold, not very productive lakes. You see higher densities in the no UV zones. So the zooplankton are actually moving down in the water column to stay out of that UV. And the highest densities were in the southern part of the lake. So here, just like looking at these graphs, these different maps, this is the sample that comes from the UV zone. So the upper part of the water column. This is no UV zone here on the right. And essentially you're looking from near shore to offshore, and the size and the color represent higher densities, like bigger circles and darker circles represent higher densities. So what you can see is that, you know, there's more zooplankton deeper in the water column than at the surface. So another thing that I talked about was veligers. So veligers can be very abundant. And so they actually tracked veligers as well. They actually didn't see, you know, a huge amounts of veligers, and especially not in the surface. So again, this is at the surface on the left, and then no UV deeper at the right. And we're seeing not a ton of veligers at the surface. There are places where they're very abundant, and they tend to be deeper. So that's the take home from that. Now, in terms of larval fish, this is what the catches of alewife look like. Alewife were the most common fish species caught. Again, this shouldn't be a surprise. This is the timing of these cruises were designed to try to catch alewife. So here, what you're looking at, again, the size of these circles represents the number. So bigger circles represent higher catches. And then the colors here represent different parts of the water column. And then, you know, it's a pie chart. So it's showing the proportion of the catch where it was caught. So here up in Manistique, they're seeing most of the catch. It was a pretty big catch, and most of the catch was in that 1% UV zone. That's different than at Sturgeon Bay, where they actually caught a bigger catch and more at the surface. So in terms of just summarizing this on a lake-wide level, most of the alewife larvae were actually captured along the western shore, which is a little surprising, and occupying the 1% UV. So they were down a little bit in the water column, not right at the surface. But this is kind of surprising, because the expectation here was that we would see a lot of fish caught around like Grand Haven, St. Joseph, Michigan City. We know this is an area where there's a lot of alewife that are produced. But again, if you remember, this sampling happened all during the month of July. But there were other people actually out collecting data separate from this, but it looked like the peak of alewife production was actually happening in June this year. So maybe, like, you know, with warming water, maybe they're actually shifting their production. We actually kind of missed the peak of alewife in the southeast part of the lake. So this is what the alewife larval fish catches looked like. It also happened to be at the same time, sort of as just a fortunate accident. We also caught, they caught a lot of larval bloater. So bloater is a native planktivore. A lot of people call them like chubs. And the bloater were the second most common species caught, 24% of the catch. And bloater were actually caught more, they were caught more offshore and tended to catch them at the surface rather than deeper in the water column. And this high catches of larval bloater seem to actually be correlated. It seems like these fish are actually survived. So it looks like there's actually a big year class of bloater that was produced in 2021 that's out in Lake Michigan right now. That actually also happens to coincide with, there was a huge year class in 2021 of Cisco and bloater and Kayai, which are similar species in Lake Superior. So there's more native prey fish out there as well. But just if you want to take it in terms of like summarizing by where the fish were in the water column, they tended to catch alewife more at the 1% UV and no UV. So alewife are deeper in the water column, whereas bloater were found more near the surface. And then if we compare that to things that we've looked at previously, There was another larval fish survey that was done during the 2015 CSMI. And what you can see there is we have a similar, the top 4 species that we're catching in these larval fish surveys are the same. Alewife, bloater, burbot, and yellow perch. But there was a much higher contribution from bloater in 2021 than in 2025. Alewife density was down. We're catching fewer alewife in 2021 than in 2015, but bloater catch increased. So in terms of the key findings, from the zooplankton community, it's a calamoid dominated system. So again, that's sort of reflective of cold, non-productive water. We're seeing high densities of veligers in some places, but not everywhere. UV radiation looks like it has an effect on community composition. Zooplankton densities are higher, deeper in the water column, so they looks like they're avoiding high UV at the surface. And alewife avoided the surface water and probably are avoiding UV exposure, whereas bloater did not. And again, larval density seems like it might be corresponding with recruitment for alewife and bloater based on the data that we've collected in 2015 and 2021. In terms of management implications, high UV, so high water clarity is altering fish behavior. Fish are lower in the water column, or it's possible that alewife are hanging out the surface are being killed. So it could be the UV as a source of alewife mortality. Is there a possible impact of changing prey taxa and the behavior of zooplankton, so that deeper distribution of zooplankton, is that influencing, that has the potential to influence alewife and bloater recruitment? And the growth and diet for alewife. So how are these fish actually growing? That's something we don't have the data yet to say. But I have a grad student who's currently working with these fish. So we have all of the larval fish that were collected in 2021, and we're looking at their diets and we're looking at their growth rates, and we're going to compare those to historical growth rates and historical diets to see if things are changing in Lake Michigan. So with that, I did want to conclude. I want to be very clear that I did not do much of this work. I'm just sort of summarizing what happened. This was a huge effort by a lot of different groups, EPA, USGS, NOAA. There's a group out of Buffalo State that does a lot of the work with the benthic surveys. If you want to learn more about this, again, we have a whole report. It's over 200 pages that summarizes all of this stuff, or well, provides all this stuff in much greater detail than what I went through today. So with that, thanks. And if I have time, I'll take questions.
Thanks, Paris.
So the 99.99, that was sort of off the cuff, but there are, so that's based on, we know, relatively, I think the stuff that Ralph's going to talk about here in a little bit, we know relatively how many fish are out there, how many eggs they produce. We know that they produce a lot of eggs and that there is a lot of mortality at that larval phase. I would say that, and then how does that, so there's always going to be a lot of mortality at the larval phase for most fish. But those differences, those like small differences, the small changes in mortality at that stage is the difference between a big year class and a small year class. The hypothesis is that mortality during that larval phase is higher now since the quagga mussels, and for the things that I tried to highlight here, like there's less food for them. Maybe it's this like changes in water clarity that maybe they're getting zapped by UV and it's killing them. Let's see. So any thoughts about doing nighttime sampling to see if the UV data holds? unfortunately, there's just, you can't do everything. Yeah. So we were sort of stuck with daytime sampling just because the different ships that were working had different protocols for how they were allowed to work. We've done some daytime versus nighttime sampling in the past, and also actually there's some data on this in this report. We do know that fish tend to be near the surface at night. So fish like zooplankton tend to come up to the surface at night. And so, yeah, they do. It does seem, whether or not they know they're avoiding UV or not, there seems to be more fish at the surface during the nighttime than there are during the day. So they're avoiding it somehow. I'm not sure that, or are they just like following the food? It's kind of hard to say. But yes, we do see higher densities of larval fish at the surface at night, but that's not the data that I'm showing here.
We have any questions from folks in the room here? I'll make a plug, kind of piggybacking on some of what Paris mentioned with the dry-scented mussels. For our April 18th workshop down in South Haven, Ashley Elgin with the NOAA Guerra Lab that he referenced out of Muskegon really specializes in the dry-scented mussels and has been working on some of the issues related to interactions with them and other invertebrates. And we'll probably give some updates too on control measures. He mentioned looking for like a silver bullet along the lines of what we have for lamprey management. And there is a product now called Sequinox that's a bacterium, a dead bacterium, kind of like BT for mosquitoes, that does kill quagga and zebra mussels and apparently not much else. So it's about as good as you can get, but unfortunately trying to apply it on the scale of Lake Michigan is basically impossible. But there have been some experiments looking at trying to do it on very localized areas.